Hi everyone. So we are on a new topic, topic 16, and we're going to talk about a couple of different case studies and a couple of different examples that have to do with phylogenetic disagreement. So what this means is that oftentimes we have cases in which different phylogenies don't agree. Those phylogenies can be from different data sets or different genes. And so we're going to talk about some of those examples. What I would like for everybody to do as part of this lecture topic is to read this additional piece of text, which is posted for you on Moodle. It has to do with primate speciation, and it's a really nice way to think about the connections between genes and speciation, because that really is what this entire topic is about. And so please read this. It will help you understand some of these other topics. Write down lots of questions that you have. And if you have any questions that relate to material from before that you might not have seen before, please ask. So the first thing that we're going to do is just to kind of lay down the framework for what we're going to be talking about the next couple of lectures. So the major question that we're going to address is how might data or trees disagree with one another? And how do systematists even deal with those disagreements? How do they do it? Are there methods? And of course, yes, there are, because people have been doing this for a long, long time. And what we're going to be doing is dividing up these topics into three different pieces. We're first going to talk about gene and species trees, which is the topic on the far right. And then we're going to talk about what happens when you've got different gene trees, so different genes giving you different answers. And then last but not least, we're going to talk about what happens if you have a morphological phylogeny and a molecular phylogeny, and they don't agree. Which one is correct, and what can you do about that? So these are our three major topics. We are going to start with gene and species trees first. So make sure that you take good notes and know where we are in the grand scheme of things in terms of these three topics, because it can get a little bit jumbled. So let's first talk about what a species tree is. All of us have been looking at trees for many, many weeks, and pretty much every tree that you have seen, either in a paper or in a lecture, is what we might call a species tree. A species tree is any kind of phylogeny that includes species as your taxa. Those species could be families or orders or genera instead, but they are still clusters or groups of species. And what I would like you all to imagine is that when we talk about species, we are really just talking about groups of populations of organisms that are interbreeding. We will get back to this topic next in about a week or so. But first, think about a species tree. Imagine that this is any tree that you have seen before. And what we're going to do is come back to this idea. So far, we've only talked about visualizing trees in terms of species relationships, but there's also a different way that we can visualize a tree. Let's first start with asexual organisms. So how might one visualize evolution in a group of asexual organisms? Asexual organisms are those that do not engage in sexual reproduction. So we might have, for example, on the left, that is a potato with little features that pop up on the potato, and those actually allow the potato to reproduce. So a lot of plants are able to reproduce by either sprouting or sending out additional rhizomes or lateral roots that allow a plant to propagate. They don't actually need pollination or additional fertilization from other plants. So they are asexual. And this is not all plants, but it does occur for many of our vegetables, such as bananas, which is actually a fruit, but you kind of get the point. In the middle is a hydra. This is an organism that is fairly simple in structure, but these guys actually produce asexually. They do not have males and females necessarily. They can just clone themselves. And then on the right, of course, is a sea star or a starfish. Those are also capable of reproducing asexually. In fact, there are many organisms that do this, but how do we visualize their evolution? If they aren't engaging in sexual reproduction, we can actually visualize their tree structures like this. So we are looking at an asexual lineage or a set of lineages, and those lineages look like a phylogenetic tree, similar to what we've seen before. In this slide, you will notice a picture of an aphid. Aphids are insects that are asexually reproducing. They can clone themselves. This doesn't apply to all aphids, but in this particular example, this is what we are envisioning. So you can imagine that we've got an aphid, some kind of ancestor, and if an aphid can clone itself, right, it can just keep producing offspring. And those offspring, those clones, are then capable of reproducing asexually without mating, and thus we get this phylogeny that is built. This is what we typically envision when we think about phylogenies, right? But here we can also think about this as the genetic lineages of these aphids. Since these are clones, their genes are all similar, and so this makes it a very simple example to visualize. Here is the phylogeny mapped out. This should look familiar to everybody. So make sure that you kind of look at this and understand what it is showing. I want you to also think about what happens when we have sexual species as an example. What might that look like? And so we're going to address that for the next couple of slides. So first, don't get overwhelmed by this slide. There's a lot going on. First, I'm going to introduce you to the term called gene tree. So we know what species trees look like, but I want you to imagine that when you zoom in on a species tree, you've actually got a bunch of different genes that are evolving within that species tree. A gene tree is the actual movement or transition of the alleles and the transmission of those alleles through time. A gene tree is basically depicting the branching events that give rise to today's alleles. In fact, if you want, you can trace back your own alleles many generations if you have that data. And if you had the genetic data, you could certainly do it, and it would be quite interesting. So what I want you to imagine here on this image, or in the image, is a time scale. We're going to start at the bottom and then work our way up. And each little pair of circles on the bottom is an individual with an allele each from their mother and from their father. Remember that you, like many other organisms, are diploid. In other words, you have a copy of each gene from your mother and from your father. Our sperm and egg cells are haploid. They contain only half the normal number of genes. So you inherit half of your makeup from your mother and half of your makeup from your father. So imagine that there are a couple of individuals at the bottom of this particular gene tree. We've got 1, 2, 3, 4, 5, 6, 7, 8. And I want you to imagine that you are the left individual. The red ball or circle is what you inherited from your mother, and the blue is what you inherited from your father. So we're going to start with you. In the next generation, you transmit one of those alleles to one offspring, and the partner next to you, the other set of circles, is who you mate with. And you have a child. You give one allele to the child, and your partner gives one allele to that child, making it a new diploid organism. So you can, if you want, take some time and you can follow up by following the lines. Each individual that will be mating with another individual in this gene pool to eventually get to the top. So over time, we will have walked through 8 different generations following 8 individuals. And remember that here we are looking at a single gene, and we're going to imagine that that gene has 2 alleles. So only imagine that we're looking at one gene and we're following those alleles through time and space. At the very top, you will notice individuals that are descendants of you, if you imagine that you are the very bottom left individual. So take a little bit of time, pause the presentation, and see if you can follow what's happening here. This, everybody, is a gene tree. Notice that this is a little bit different from a species tree because you're tracing individuals. You are not tracing species groups over time. Here we have the same gene tree presented, but what's happened here is in this particular presentation, we're going to follow one particular allele, and we're only following it in one direction. What we start with there is the 4th individual on the bottom. So that is the oldest, the ancestral allele, and we're going to follow it to its right. The double line there should just be a solid line, but you can imagine that we've got that allele that gets transmitted to an offspring, one individual, and then from there on, generation through generation gets passed down to other offspring. So this is a simple demonstration of an allele. We're only picking one, and we're going to follow it through 8 different generations to the very top. What you can imagine is that those individuals at the very top initially had an ancestor and an allele in that ancestor that can be traced back. So very much like a species tree, we can actually trace alleles back through time. Let me introduce to you a couple of terms that we're going to use for this topic. First, you will notice that all alleles can be traced back to a common ancestor. You yourself could trace your alleles back many, many generations to your ancestors, which is what we do when we engage in activities with, say, 23andMe and Ancestry.com, right? All alleles can be traced back to a common ancestor, and this is called coalescence. Coalescence basically means coming together, right? If you follow the black dots with the black lines all the way to the base of the tree, or even to the 2nd generation, those alleles coalesce there. That is where they meet. The reason that this particular allele doesn't go all the way to the very 1st generation is because the 1st generation where those alleles come together is the coalescence point. All right, so coalescence is the coming together of an allele, starting at the top and working your way back through the generations. Another term that I want you to get familiar with is something called lineage sorting. Sometimes gene lineages or allele lineages are lost over time. I'm going to point out an example here in the slide, and you will notice that here, this particular allele doesn't go any farther. That is the end for that allele in that generation, in that particular individual. And it could just be that that individual allele isn't the one that gets used or gets transmitted to the offspring that comes next. Or you might also imagine that an individual just doesn't reproduce and doesn't mate. Something else that I want you to remember is that some gene lineages or allele lineages can die, but no new gene lineages can appear. Genes do not spontaneously arise. However, every now and then, new mutations can pop up in a population. We're going to pretend that that doesn't exist in this example. All right, so get familiar with the term coalescence and lineage sorting. So here's another example. And what I want you all to imagine here is that all of these different alleles, whether it's the black or the white one, can be traced to an ancestor. For the ease of this particular example, we are going to trace the black allele from the top, from the species, or individuals, if you will, A, B, C, D, E, and F, down to where that allele or gene coalesces. Coalescence, of course, is where the alleles meet. Where do they first diverge? And so we can actually trace the black allele all the way back through these 8 different generations to the very base of this gene tree. A fairly simple example. Now, if you were to draw that particular phylogeny and those relationships, we would get something that looks like this. Imagine that this is one gene. One gene, 2 alleles, and this is what we get. Species A and C actually share a most recent common allelic ancestor. See if you can follow that through. Then, after that, B and D are sisters in terms of their allelic makeup. If you follow back all the way to the 3rd generation, A, C, B, and D meet. And then we can do the same for E and F. E and F go all the way back. They coalesce in the 2nd generation. And if we follow all these individuals, A, C, B, D, E, and F, they coalesce in that very 1st generation. So imagine that this is one gene. Now let's imagine that we've got a separate gene that we are looking at. Remember, these are the same individuals. They all have the same genes. But remember, you have thousands of different genes, so each gene can be traced separately. Here, we perhaps have a different gene tree. So we've got individuals A, B, C, D, E, and F. Let's see if you can figure out where these coalesce. So pause the video and see what you can come up with. Now let's imagine that we want to draw this in a tree shape or in a tree form. What might that look like? You can again pause the video and see if you can figure out how that might work. Don't let the black lines confuse you. Just follow those alleles back to where they coalesce. Here is the gene tree that you might get, or the gene phylogeny that you might get for this particular gene, and you will notice that if you compare the 2 trees from gene 1 and gene 2, they happen to be very, very different. This particular example and this phenomenon is called gene discordance, or discordant genes, or discordant genealogies. These 2 different genes are telling us 2 different stories. That is very interesting. We would expect that the genome, which is the combination of all genes for an organism, would say the same thing, but in fact they do not. So why does genealogical discordance exist? Why is it that 2 genes are not necessarily telling the same story? And without going into too much detail with genetics and genetics concepts, I'm just going to give a basic overview. If you really want to know the details, we can discuss these in our group chat. But here is what I want you to remember. So if you think about a set of chromosomes, or just a chromosome in general, you have several different genes located on that chromosome. We call those loci. Distantly located genes, or loci, may track a different evolutionary history, even though they are on the same chromosome. And that is because of recombination. If you remember, going back to genetics or some basic biology that you've learned, perhaps even in high school, there is something that happens during the process of meiosis, which is when we are producing sperm and eggs, that causes genes to be shuffled. I will show an animation here in a moment. Any genes that are adjacent to one another, so those genes that are close on a chromosome, tend to track the same evolutionary history, and I will show you why. Finally, some genes actually evolve more rapidly than others. So you can imagine that different genes might tell you a different story about either the age of a clade or a group, or some genes might track different histories because they have literally evolved differently. And so let's take a look at this quick little animation. Here, we're going to start back from scratch. This is the process of meiosis. There you've got the centrosomes moving to the opposite sides of the cell. Homologous chromosomes line up, get pulled apart, but you will notice something happens before those chromosomes get pulled apart. So let's finish this demonstration and we'll start over again. Here are chromosomes. There's only 2 are lined up. They line up and they happen to swap parts of their genes on those chromosomes. That is called crossing over, and the result of that is recombination. With every meiotic event, so meiosis, you are essentially creating a new combination of genes on a chromosome. So if you'll take a look at this little animation, you will notice that there are going to be some genes that are swapped in those chromosomes. And what that usually results in is that some genes are going to follow a different evolutionary trajectory than others will. You will also notice that if genes are located more closely to one another, they will probably end up swapping together or not. Here's another way of looking at this particular phenomenon. This is just a random chromosome number 19, and you will notice that there are different colored bars on this chromosome. Those are all different genes or loci. And what you will notice is that you can pick any random group of loci, as has been done in this image, and you might get a different phylogeny, a different set of relationships for each of those different genes. That is because different genes are undergoing different evolutionary trajectories, depending on whether they have been recombined, and it also might have something to do with the rate at which things evolve. I will also show you some other ways in which these phylogenies might appear a little bit differently. Let's take a look at how gene trees and species trees come together. So this is how we might be able to solve the problem of having different gene trees and potentially different species trees. So again, I want you to imagine that a species is just a group of individuals in the same population. If you look at the light gray color, that is our species or population tree. So we've got species A, B, and C, or you can think of it as population A, B, and C. Species A, B, and C have over time undergone some speciation events or some splits. And if you think back to some of your other biology courses, there are a couple of different ways in which populations can split. So let's start with this first question. Where do species or populations A, B, and C coalesce? I'm going to give you a moment to think about that. You can pause the presentation and see if you can answer that question. Where do A, B, and C coalesce? All right, very good. A, B, and C coalesce at the point in which all of them come together at a node. Now, one thing that I want you to notice is that you've got the gray tree, which is your species or population tree. The gene tree is actually what the genes are doing, and this is for one gene at a time. So the gene for A, B, and C coalesces at this particular node. If you look at the species tree, it looks very similar to the gene tree. In fact, they are identical. The reasons for species splitting over time could be due to speciation events that are the result of a geographic barrier that divides a population into two. So for example, B and C at some point shared a common ancestor. They were one population, but they got split over time due to some geographic barrier, such as a mountain range forming in between them or a river forming in between them. Think of something like the Grand Canyon, which formed over many, many, many years, and that happened because of the Colorado River eroding those rocks away. And so you actually have different populations of organisms like squirrels that shared a common ancestor at some point, right? So at split B, species B and C, or populations B and C, actually split due to some geographic barrier or some other speciation event. You can also follow A, B, and C down to split A. At some point in evolutionary history, species or population A split from another population, which eventually became B and C. And here's a second question. Where do B and C coalesce? That is fairly easy to see. Here, the gene and the species trees are concordant. They agree with one another. But what happens if they don't? So remember again that the gray shaded parts here are the species tree or the population tree, and the black lines are the gene tree, or represent the gene tree. So what is happening here is that species B and C actually don't really split genetically. Compare that to species A, B, and C. Species A and B actually split at point A, but species B and C do not split genetically at split B. This is a phenomenon that we call deep coalescence. For some reason, species B and C don't really separate into two distinct species genetically until perhaps much later than you would expect. You would expect that at split B, species B and C would split and be two genetically different entities, but they do not. And in fact, if you go back far enough, species B and C don't coalesce until the very base of the tree. That is where they coalesce, whereas species A, B, and C coalesce at the same point. Deep coalescence basically exists because at some point in time, populations B and C could have been intermixing. Perhaps they came back together and started interbreeding again. Or perhaps there was some gene flow between the two populations. Here is a blue bracket that illustrates the point during split A and B where species B and C did not split. Instead, those two lineages are still evolving. And at the same time, the ancestral alleles for B and C still coexist. They don't disappear. Here is another demonstration of this particular phenomenon. Let's see if we can trace back for species A, B, and C, or populations A, B, and C, their alleles, and see where they coalesce. So this is a different demonstration. This is actually the gene tree, and you can imagine that all the genes combined are going to form the species tree. So let's take species A, which has the little black allele, and we can trace it from those 4 individuals all the way back to the 2nd generation. If we take species B and we trace its light gray circle or allele all the way back, it coalesces with species A in the 4th generation. Now let's take species C with the darker gray circles, and it coalesces with those 2 species in the 2nd generation. All alleles coalesce to an ancestral allele, so there is coalescence. The B and C gene lineages are retained between the 2 splits A and B. This is called incomplete lineage sorting. Go back to how we defined lineage sorting and make sure that you understand this concept. Incomplete lineage sorting is when alleles do not go to fixation, or alleles do not disappear over time. So here, during split A and split B, we actually have both the alleles for B and C maintained at the same time, and they don't coalesce until the 2nd generation. So B and C coalesce at the point where A, B, and C come together. So which relationship should the species tree indicate? Don't think about the genes, just think about the species tree. In order to answer this question, think about how speciation occurs. Speciation occurs due to a split in the population, so the species tree should be illustrated as B and C having a close sister relationship, followed by A. Which relationship should the gene tree indicate based on what we have just observed? Are B and C still sister taxa, or does this look a little bit different? Instead, here, A and B actually share a more recent dissension point or coalescence point, as opposed to B and C. And if you all remember, going back to some of the very first material that we covered, how do we estimate relationships? Remember that relationships are based on recency of common ancestry. Since A and B coalesce more recently, they should actually be sister taxa. That is what the gene tree is telling us. So what we have observed here is something called deep coalescence due to incomplete lineage sorting. The alleles that belong to B and C still exist between split A and split B. So this is a demonstration of how gene trees do not always agree with their species trees. There are ways to reconcile the two. There is software that you can use to determine how your gene trees and your species trees fit together. We will not be talking about those methods. They get quite complicated.